Hello, everyone. Some familiar people have signed on here. Welcome, everybody, to the February virtual brown bag here at the Center for Science of Information. With us here today is Mainak Chowdhury from Stanford. And here at the center, we tried to come at diversity at a number of different ways. And so we work to include a brown bag that really stretches the kind of application of science for information in unexpected ways. And the idea is that when we think of diversity more broadly, it helps us to become more accustomed to diversity in a more traditional sense. So Mainak is going to talk to us about the intersection of science and information and clinical care applications for cancer and rare diseases. And if you haven't already, before I turn it over to Mainak, down here is a poll. If you could sign in there, you'll notice down there a place where you can type in questions. We will save some time at the end of the talk to take more questions. And with that, I think I'll hand it over to you, Mainak.
Thanks, Kelly, for the introduction. Good morning, everyone here on the West Coast, and good afternoon, everyone on the East or in the center. So today I'm going to talk about our work on making information sciences accessible to clinical care providers in helping them make better treatment decisions. As you can see from the title, my narrative will focus on cancer, but I want to emphasize here that this work is not restricted to just that. This is joint work with Frank Rivelbis at Purdue. Okay, so we start with an unfortunate reality about cancer treatment today. Even though cancer accounts for around 500,000 deaths every year in the United States and is second only to heart diseases in its prevalence, the exact molecular or the biological mechanisms behind cancer is not known in general. There are some specific cases where we know a great deal more than in others, but the overall state of affairs still remains that we do not understand what cancer is caused by and how it develops. In other words, cancer itself manifests in a variety of forms. Identifying different forms of different stages of carcinogenesis and their triggers and suppressors, that continues to be an active area of research. In particular, even though the superficial symptoms may be the same for different cancers, the underlying causes can be vastly different. And this is not only across different people. This is what we mean by different, by the heterogeneity in the patient population. But even in a single person, there may be different microenvironments at different sites of cancer. So this particular figure here is taken from a paper in Nature Reviews, and it talks about this aspect in terms of non-small cell lung cancer. And it makes the point that the tumor microenvironment has important roles in determining the characteristics of lung tumors. It is likely that a combination of the cell of origin, the genetic alterations, and the microenvironmental factors all contribute to the lineage identity of lung tumors. And this is verbatim from the text, from the paper. So as you can see, that there's a lot of, even though cancer is a very, I would say, prevalent disease, but any specific kind of cancer is still a rare disease, which is why we had cancer and other rare diseases in our title. Furthermore, new research, which we do not cite here in these slides, based on some new imaging techniques, for example, single cell imaging, there is a new line of work which suggests that cancer development is not just genetic in its origin. There are factors behind the development of cancer which cannot be predicted entirely from genes, from the genetic makeup. Uh, it depends on other epigenetic factors also. Um, so in the absence of a definite understanding behind the mechanisms of cancer, most of the common treatment options today are mainly validated by trials on patient populations. Clinical trials, however, are really expensive. So currently, and this figure is current as of 2 years ago, 2 or 3 years ago, we have close to 150 to 200 FDA approved chemotherapy drugs. And the reason this number is so low is that new drug discovery is really expensive. Each new drug costs upwards of billions of dollars. So considering this unfortunate reality, combinations of drugs or something known as combination therapy is a standard protocol that is followed by many doctors when it comes to clinical care of cancer. And this is true not only for cancer, but also other, let's say, infectious diseases, where antibiotics, for example, would or bacteria would develop resistance to certain kinds of antibiotics, and then one would often have to resort to trying out different combinations of antibiotics, or trying out a stronger antibiotic, and not wait for the cost of developing an entirely new antibiotic altogether. And the reason behind why this approach is a viable technique, or has proven to be or continues to be used and trusted by lots of clinical care providers, is that at its core, combination therapy essentially relies on exploiting the synergy in the administered drugs. So even though the drug was not specifically meant, or let's say clinically or through clinical trials tested for a particular application, it may be that doctors, through their judgment, can decide or can see that certain combinations of drugs would help each other's effects, and thereby treat the overall disease much more effectively than if any one drug were to be used in isolation. However, it still remains, the fact still remains that cancer, we do not know how cancer develops and what it's due to. And this manifests itself in a lot of problems. First of all, it continues to be the case that most chemotherapy regimens are hit or miss. It either works or it does not. It's fine if it works, and the way we quantify whether it works or not is through observing whether there is a relapse of cancer, or whether after treatment the cancer cells grow back or not. So while there have been success stories associated with chemotherapy, the overall landscape is still, I would say, dotted with lots of unfortunate stories about drugs not working when we thought that they would. And the reason why this is bad is that generic chemotherapy drugs, they have serious side effects. So not only does not working, not only does the patient lose time, but the patient also undergoes some serious side effects due to wrong treatment, or drugs that were not suited for him or her in the first place. In particular, a particular drug that works for one patient may not work for a different patient. And even in one patient, what works in a particular microenvironment may not be that effective in a different microenvironment. And testing all of this across all possible variations of microenvironment and patient populations, this is an expensive proposition. And clinical trials are, in fact, if you look at the breakdown of costs associated with discovering a new drug, the clinical trials form a bulk of the overall cost involved in drug discovery, upwards of billions of dollars. So the starting motivation behind our project is that we would not like to rely solely on just the clinical trials for assessing the effectiveness of drugs. We would want to make sure that whenever a doctor administers a certain drug or a combination of drugs, we would like to understand what the records of the treatment administered and the eventual outcome that was observed, what that has to say about treatment options for a different patient. In other words, we would like to extract clinical value from existing records of administered treatment and treatment outcome. So just to reiterate, our long-term goal is to enable extraction of clinical value from records of treatment outcomes. Now, as you can imagine, acquisition of this data set that we just talked about of administered treatment and the eventual outcomes, that's a very tricky proposition. So I have here two pictures. One of them are binders of medical records, which is just to remind ourselves that even in this day and age, you would be surprised to find out that many record transfers among hospitals, they take place not electronically but via printed sheets. So if you want to transfer, for example, patient records from one hospital to another, again, there are exceptions to this, but there exist cases when people would literally print out records from one hospital and then they would manually enter those records in a different record keeping system. So there's a lot of fragmentation in data sharing amongst healthcare providers, and this is in fact an unfortunate reality that would impede let's say fluid access to data sets from different healthcare providers, and the reasons are many. So there are concerns due to privacy. There's not enough incentive for people, for healthcare providers to share data. There are competition issues. It takes it's costly to acquire this data set, and it's not clear what financial incentives healthcare providers would have in just giving this data set away for free. Also, from an information analysis point of view, there is no standard for the data acquisition. Again, this is something that is changing as we speak now. There is a lot of effort to make things more standardized. There are lots of both private and government initiatives in trying to standardize data transfer amongst healthcare institutions. But the reality today is still that it's an ongoing work. It's still work in progress. In fact, the importance of data sharing has been recognized in the latest initiative announced by President Obama in the beginning of the year. Among other things, it emphasizes on the importance of data sharing to make sure that we have all the tools available to us in our fight against cancer. So it's no longer an option for us to keep our data and our common knowledge away from each other. That's no longer an option for us. So all of this would take time for the data to be accessible. For the duration of our project, we cannot afford to wait that long. So what we decided to do instead is to get our hands on the first publicly available data set that we have and try to explore how we can meaningfully extract clinical value from such a data set. And in the next slide, I'll talk about the data set that we end up choosing. So this is the multiple causes of death data set, which is a record of all cases of mortality recorded in the United States. So each record is one person, and roughly there are approximately 2.5 million records per year. This corresponds to the number of deaths in the United States per year, and each record contains conditions that existed at the time of mortality, and these conditions include demographic information such as race, age, education status, or place and time of death, and a list of clinical conditions. For example, what conditions was the person suffering from when at the time of mortality, and so on and so forth. And the most important thing about this database for our purposes was that this dataset was open for research and academic uses. So we decided that this would be a great starting point for our endeavor to enable value extraction in terms of association rule learning among the different conditions that exist in the multiple causes of death dataset. Now, what do I mean by association rule learning? So I'll come to that in the next few slides. But before that, I would like to draw your attention to user assisted. So this is one way in which we see our endeavor as being different from many other initiatives by, I would say, mostly coming from the machine learning community, which is trying to let the machine itself mine through lots of data and trying to come up with recommendations. We believe that that is a very valuable approach, but at the same time, there is value to a person with a domain-specific knowledge contributing or being able to contribute his or her domain expertise to enriching data sets or enriching the value that can be extracted from data sets. So just to give an analogy, it's the same kind of thinking that we feel is relevant between an initiative like Microsoft Encarta, which is a private initiative to build an encyclopedia, and Wikipedia, which is a public initiative to build an encyclopedia. And we feel that the approach taken by Wikipedia is more sustainable in the long run, and this is what we are inspired by when we try to build this platform. All right, so now we talk about, this is just for illustration. This is a typical record in the multiple causes of death file. So this record corresponds to the mortality for one person, and as you can see, it has different codes associated with different conditions. For example, this highlighted portion here, F329, this is something called the ICD-10 code, ICD-10 being the International Classification of Diseases, 10th revision code for the primary cause of mortality for that person. So together with the primary cause of mortality, there are also secondary conditions that existed at the time of mortality. And what we would like to do based on this data set is we would like to find out what conditions are associated with each other. And this is something that is well known in the machine learning community as something known as an association rule learning. So if you are familiar with online marketplaces like Amazon or online recommendation systems like Netflix. Um, association rule learning, also known as market basket analysis, comes up very often. So what you would like to do in such a situation is, given the relative frequencies of different combinations of conditions, or in terms of markets items, you would like to find out items or conditions which appear together, or for which there are significant statistical, I would say, measures of relevance. So in particular, to keep our to summarize the gist of what extracting a rule implies, we would like, when we say we have found a rule, we would like to make connections like, given condition A and condition B, condition C is most likely to be there, or is likely to be there too. So A, B and C appear together. So this is something we would like to be able to extract from the database with an objective measure of certainty. Now, as you can imagine, extracting, there are first of all many such rules that are evident or that can be justified using the data set according to different statistical criteria. And this is the approach that the machine learning community is taking, as in what kind of statistical criteria would lead to relevant rules, or rules that identify patterns that were not known before. What we do here is we try to enable an user with a domain specific knowledge about the specific conditions to be able to inform this rule identification and filtering. We would like their input to inform better decision making by our system, and in doing so we would hope that this would be a platform where which would learn from the users and which can clarify doubts with the users if it is not certain about the effectiveness or about the validity of a certain rule. All right, so now we'll do a quick walkthrough through what a prototype of our interface would look like. So as a working example, let's explore conditions that one would associate with the most common type of lung cancer. And what this would entail is that the user would enter a particular condition, lung cancer. The system would return several conditions that are associated with that. The user would select conditions that he or she is interested in, and the interface would subsequently return related conditions, and so on and so forth. Eventually, the user would be able to contribute discovered patterns back to the system, so that that can benefit others who are searching for related queries. Okay, so that was a very high level description. So we'll do a quick walkthrough. I'm not sure how visible this is, but what we see here is essentially all the conditions that appear in the multiple causes of death dataset across the last 6 years. So all that data is summarized in this list that we see at the bottom here, and the bubbles that we see in this space right here is associated with each such condition that is listed. So there are 9 bubbles here for the top 9 conditions. So this is a visual way of seeing what is the most commonly occurring condition across the MCT dataset. So once the user sees this, he or she has the flexibility to choose a particular condition or filter the list of displayed conditions by typing in keywords. So for example, so in this particular example, we want to explore lung cancer. So we type in neoplasm or ICD-10 neoplasm. And this automatically filters the list to reveal, again, different cancers by their frequency. And I'm not sure if this is very distinct here, but the biggest bubble here, this represents lung cancer. And it has a very specific code, which is ICD-10 C349, which is malignant neoplasm of the bronchus or lung, which is unspecified otherwise. And this happens to be the most common occurrence of lung cancer across all the across all the MCD dataset across the last 6 years. Okay, so we want to find out what the associated conditions are. So we go ahead and select this condition, which is lung cancer, and this is indicated in cyan here. And then we ask the system to list related conditions. And once we do that, we see exactly what we see on the bottom half of the screen here. Again, this is, I apologize for this not being very distinct, but as you can see, we have a list of different conditions here. So for example, the most frequent condition that is associated with this specific type of lung cancer, or this most common type of lung cancer, is mental and behavioral disorders due to the use of tobacco. So you can right away guess that smoking or tobacco use is the most common, and this is probably not coming as any sort of surprise, but tobacco use is associated with lung cancer, and it's the most common, I would say. It has a high correlation with occurrences of lung cancer. Also, if we look down further, we would start seeing, for example, conditions like heart disease, different kind of respiratory failures, pneumonia, and this is not here in the list here, but if we scroll further down, we also see complications in the kidney, for example, due to the use of that is associated with this particular form of lung cancer. So this specific examples might suggest that the drugs that are used to treat lung cancer, they may have some adverse effects on the kidney, and this is something that we can extract right from the available data without having let's say any idea of what the drugs are or what is it that actually causes, or what are the molecular mechanisms behind, for example, those complications. What we have here instead is just a very objective statement about the fact that this complication shows a high degree of correlation with lung cancer, and that is what we would like to enable. So this particular screenshot, it shows again what happens once we select a particular condition. So in this particular example, we select essential primary hypertension as the condition that we would like to explore further. And once again, the system returns the list of all conditions that are associated with these two conditions. So the list in the bottom half of the screen now, it shows all conditions that are associated with both these occurrences, lung cancer and primary essential hypertension. And we could go on building trees like this. And once we have found, or the user has found, a significant pattern, he or she can upload this to our system and mark it so that it can be used for further association rule discovery. this is something that is currently a work in progress, and we hope to have that up soon. Okay, in this slide, I mentioned some algorithmic issues that we had to deal with while we were designing this system. So the size that we are dealing with of the data set is roughly tens of millions. So it's roughly 2.5 million per year, and we eventually intend to use all the MCT datasets that are available, and MCT datasets are available all the way going back to 1979. So we can, so the size of the data we are talking about is on the order of hundreds of millions, with the total number of conditions being around tens of thousands, and the conditions can be both the ones that are specified in the multiple causes of death data set, as well as conditions that the users would specify afterwards. And as we saw, each record, it has roughly 10 to 50 conditions per person. So that's the size of the data set that we are talking about. And at each stage, when the user selects a particular list of conditions, what the system does is the system has to, in some form or the other, compute some measure of statistical relevance. So in this particular case, we compute the relative frequencies of occurrences of combinations of those conditions across these tens of millions or hundreds of millions of records that we have for all the MCT files. After having done that computation, we would like to filter them based on whatever the user specifies as being interesting, and then incorporate that in a visualization that brings out the significance of the data. And that is all done by the system. And this is something, again, that I indicated that this is still a work in progress. So right now we are able to specify only a limited set of conditions. We are working on ways to let users specify much broader sets of conditions and enable pattern discovery or association rule discovery with the broader set of user-defined conditions. And we expect that this flexibility would be invaluable to helping or to enabling a domain-specific expert to identify patterns that already exist in data, instead of letting the machine do all the guesswork. Okay, just to summarize the work in progress. So we are exploring new criteria for filtering information. So we want to be able to return conditions that the users would find most interesting. And in doing so, there are different statistical criteria that we could choose for the purposes of this demonstration that we saw just now. We used relative frequency as a measure. So the most frequent conditions or the conditions which appear with the highest probability, those would be the ones that would be displayed at the top of the list. But we could choose other measures also, and this is something that that we are looking into. Also, we would like to integrate user contributed descriptions of conditions. We believe that this would be very critical in helping users derive value from our data set, from the data set that is already publicly available. Also, this particular data set, while we started off with this, we believe that down the road we would need to integrate data from multiple other sources also. In particular, there are lots of public data sets which associate genotypic and phenotypic responses to particular drugs, for example. And these data are highly relevant to what we are trying to achieve here. And we believe that association rule learning should also exploit the information that is there in the other data sets. With that, we come to an end of the presentation. I'd like to take this opportunity to thank Brent for coordinating last year's data science workshop, which started this entire project. So thank you, Brent, for putting this together, and I'm glad that we got an opportunity to attend the workshop and develop this idea further. Thanks to all members who helped out with the workshop also. Luke in particular was very helpful with helping us with the refinement of our ideas from the point of view of the end user, as in what is it that would be really useful to doctors, for example, in what we are doing. Luke was critical to helping us understand that better. Also, we would like to thank Dr. Ward and Dr. Haugler for helping us with the workshop and for exposing us to ideas related to both data science techniques and to techniques that are commonly available in biology labs. And these techniques are useful, are very helpful to our understanding when it comes to understanding drug interactions, for example. And again, we would like to thank the Center for Science of Information for providing us financial support in the form of travel grants, which were very helpful in scaling up our implementation to real data. So I cannot emphasize enough how much of a flexibility that gave us in helping us scale the scope of data that our platform would be able to process and analyze. So with that, come to an end of my presentation. Thanks all for your attention, and if you would have any comments or questions, I'd be happy to take them now.
Well, if no one has any questions, I will thank you again, Mainak. Thank you everyone for tuning in. Okay, great. Thank you so much.
Thanks a lot, Kelly, and everyone for attending.